Hello everyone, welcome to QXon. In this video, I will talk about methods used to demonstrate the functions of cells and its products and its communications, and what are the applications of these methods in clinical care and research. So till now in general physiology we have seen that cells have a shape and a structure, then different organelles of cells perform different functions. Then cells have a resting membrane potential and they respond to a stimulus by generating action potential or by causing contraction. Then cells secrete also and they have some intracellular contents, plus they express protein on their membrane intracellularly, and some of these proteins are secreted as well. So if we know the basic structure and function of cell, from that we can know what to assess. So we can find out that are size or shape of cells normal, is the gene function normal, are the organelles present in the cell functioning normally or not, then the intracellular proteins and the surface proteins, are they expressed normally or not, or are they deficient. Then the electrical activity of cells, suppose neurons or muscle, is it normal or not. Then are secretions of the cell, they are really being secreted or not, so we can assess in blood whether they, what is their concentration. Then are cells damaged and their contents released into circulation, so abnormal secretions which are present in circulation, that will also give an indication of the functioning of the cell. So first method by which we can analyze the function of the cells is that we can actually take out the cells. So we can use various methods for taking it out, suppose scraping. So suppose if there is a skin lesion, we can scrape the surface and get some cells from that. Then there is fine needle aspiration cytology, that is FNAC, from that also we can get cells. Then biopsy, so we can cut a sample of the tissue from the affected tissue and we can analyze it. Then we can also take a bone marrow sample, we can insert a needle into the bone and we can get a sample of the bone marrow. Similarly, we can get the samples of body fluids, so we can get a blood sample or urine sample, and further we can stain the sample and look under the microscope. So what has affected the cell, that we can directly visualize under the microscope. So we can see the shape and size of the cell. Suppose there is anemia, right? And if we take out a blood sample and stain it with some stain and look it under the microscope, we can actually see the shape and size of red blood cells. So if suppose there is sickle cell anemia, the cells will appear sickle shaped. So just by directly looking under the microscope, we can know what has affected the cells. Then we can also look for the presence of inflammation, whether it's acute inflammation or a chronic inflammation. So if it is acute inflammation, we will see the presence of neutrophils along with the cells which are there, and we can see that some areas have also been damaged by the due to inflammation. So that we can directly visualize under microscope. Then the same sample we can use antibodies to label the antigens, which is nothing but the proteins. And this process of labeling antigens with antibodies is known as immunohistochemistry. So this will help us in identifying presence of abnormal antigens and study normal tissue and cells. So suppose this is the sample which we have taken, and if there is a presence of protein, we don't know whether the protein is expressed or not. What we do is we add a primary antibody, which if the protein is present, it will react with that protein, and then we add a labeled secondary antibody. Labeled means that we can actually visualize under the microscope. So there are various methods of that, we will not go into detail. We can actually see if we can actually tag it with some enzyme and see the product which has formed. So if the labeled secondary antibody combines with the primary antibody, we will see the presence of the colored product. So, it will help us in identifying the presence of protein. On the other hand, if there is no protein, then this reaction will not take place. So, this method of identifying the presence of antigens or proteins on the surface of the cell is used to study normal tissue and cells also. So, it is a very good method for identifying the expression of proteins on certain cells and is used extensively in research, and it also helps in identifying presence of any abnormal antigens present on the surface of the cells. So, in summary, just take out the cells by different methods, stain and look under the microscope, or we can use antibodies to label antigens, a process known as immunohistochemistry. Then we can actually assess the functions of various organelles. So, let us first consider the function of the nucleus, how it is assessed. So, for that we can do actually karyotyping. With that, we can find out the number of the chromosomes with the characteristic staining pattern which we get, and we can actually visualize the chromosomes after staining. Then there is a PCR, or what is known as polymerase chain reaction, and then there are various blots. So, there is southern blot for DNA sequence, then northern blot for studying mRNA, which is formed when the genome is transcribed to mRNA. Then western blotting is done for proteins, and then finally we have eastern blot, which is to study the post-translational modification of the proteins. So, it is like from DNA mRNA is synthesized, and from mRNA proteins is synthesized, and then after synthesis there is some post-translational modification of proteins going on. So, all these aspects can be studied using different blots.